briefly introduce our speaker today. That's Professor Zhu Shiwei from our physics department. And he got his PhD in 2004 from the photonics and optoelectronics at National Taiwan University here. And 2 years later, 2006, he joined the physics faculty of NTU, our faculty. And about 12 years later, 8 years later, in 2014, he was promoted to a full professor. And his research is basically aimed to explore the boundary of optical microscopy with applications on biology and material sciences. And about the awards, he received Young Scholars Creativity Awards of the Foundation for Advancement of Outstanding Scholarship, the Outstanding Young Scholar Research Project Award from MOST, the Ministry of Science and Technology, and the MOST Future Technology Award. And currently, he serves as an associate editor in biological reports and also a board member of Taiwan Physical Society. Apart from his own achievements, his students actually, under his supervision, also have won more than 60 research awards. So for the time being, so far, Professor Zhu has devoted a lot of time on not only on research, but also on education to have won him numerous teaching awards, including the NTU Outstanding Teaching Award and the Excellent Mentor Award. So today he's going to tell us about some mystery or mysterious secret about the human brain. So let's welcome Professor Zhu.
Yeah.
Right, the floor is yours. Yeah.
Okay, yeah, thank you so much. So the voice is clear? I hope it's so. The voice is clear now, like this. Hello? Anyone hear me?
It's very good.
Very good. Thank you so much. Yeah. So yeah, I appreciate Junhui's kind invitation last week because we have a speaker who is not able to come here. So Junhui would like to have a colloquial talk specifically about biophysics. So I kind of volunteer to give this talk on our research results on brain functional imaging with light.
Yeah, I forgot to mention that a lot of appreciation that Professor Zhu actually accepted this invitation with a very short note. And I think basically what he's talking about today is kind of perfectly fitted into the need of this colloquial series. Right. So yeah, carry on.
Okay, yeah. So I appreciate to have this opportunity to share our recent research progress with our own faculty members as well as with our own students. And I hope this plays a stimulating example for you to also consider joining this research direction on brain. I think I should start following the talk of Professor Guo Shangzhi. Last week, the aim of his talk was really to motivate students toward quantum science. And I think my aim of today's talk is really to stimulate our students as well as faculty members' interest to brain research. So as Junhui mentioned, I'm mainly affiliated with NTU Physics, but I'm also affiliated with NTU D School, where there are a lot of interdisciplinary professors in NTU gathered together, and I really enjoy discussing and teaching there. Also, I'm affiliated with Molecular Imaging Center because I work a lot on molecular imaging with light, as well as National Tsinghua University Brain Research Center. So these are my few affiliations, and I really enjoy this kind of interdisciplinary and the discussion with many different fields of researchers. So to begin today's talk, I would like to start from these two articles. One is in Chinese, one is in English. So in case you missed any content that I'm going to talk today, because today's online presentation and it's very difficult to concentrate online for an hour. So on one side, I will try to finish my talk as soon as possible. On the other side, if you missed any content, you're welcome to refer to these two papers where all the technical content that I refer to will be included in have been included in these two articles. Okay, so I would like to start from the concept of golden circle, which was proposed by Simon Sinek in a famous TED Talk 10 years ago. And this is actually talks about how to consider research. When we talk about research, very often we start from what? Especially when we talk with students, we talk about what we are doing. But it is really most important to start talking about why this research is important and how we want to carry out the research. And the last part is what do we obtain in the research. So I would like to follow this golden circle sequence and lay out my talk today as why starting the brain, and in terms of how we choose to use light microscopy to study the brain. And I will explain why we choose light microscopy and for what. I will tell you a recent result on achieving functional connectome observation for individual neuron in a whole intact in vivo brain. So this will be my outline today. So I would like to start from why studying brain. I think the motivation should be obvious that everyone has a brain, including you and me. While you are listening to my talk, your brain is functioning, your brain is thinking, partly might be on this talk, maybe partly on other stuff. So we all have a brain, but how does it function? I think none of us really understand, right? So I think this is the most fundamental motivation. In terms of physics, we hope to understand our natural world. So brain is one important component of the natural world, one important component of our body. So to understand brain is really a fundamental issue in terms of physics. And to give a stronger motivation, to give a broader interest, I would like to cite this Science Spatial issue in 2005. And in this issue, this was the 125th anniversary of Science. And in that issue, this journal invite many renowned researchers around the world and summarize 125 important questions that we human beings are probably able to resolve in 21st century. Okay, so within top 25, the second important question is what is the biological basis of consciousness? And I think this is about 15. How are memories stored and retrieved? These are all relevant to brain science. And since I'm talking to physics students mostly, I would like to invite you to think about, to make a guess, what is the number one question? Unfortunately, today, this is an online format. Initially, I hope to see all of you and ask you this question. But anyway, please think about this and make a guess. What is probably the number one question out of these 125 questions? And I give you 5 seconds to make a guess. And the result is, in that issue, the number one question is, what is the universe made of? I think this is actually a perfect fit for physics students. I mean, similar to Professor Guo Shangzi's talk last week, when people talk about quantum science, suddenly physics become one of the core discipline in quantum science. And I hope physics students feel privileged to join this quantum science research. Similarly, the number one question in this science spatial issue is, what is the universe made of? This is a very typical question that we as a physics faculty ask all the time. Okay, so but today I'm not going to address anything on this universe made of issue. I'm going to talk about mainly on brain, mainly on the biological basis of consciousness and as well as memories. Okay, so again, to follow Professor Guo Shangzi's style, he actually gave a map last week and highlighting that, all over the world, there are a lot of quantum investment. And very similarly, all over the world, there are a lot of brain project in the world, so including Europe, States, China, Japan, Australia, and we in Taiwan also have recently we also initiated a BRAIN project. Even though the funding size, I don't have a real detailed funding size as Professor Guo did last week. I didn't spend 200,000 NTD to purchase that document. But anyway, we do have a BRAIN project in Taiwan, even though I think the budget size is kind of one order smaller compared to Europe and in the States, and as well as China's investment. But we do have a specialty in brain science, as I will reveal to you later. Taiwan do have a spatial strengths in brain science all over the world. Okay. So I would like to start from the introduction of brain science in general for our physics faculty and students. So if we think about brain science, well, human being is curious on how brain functions, not only now, not only 100 years ago, but a very, very old problem, a very old question. So we may consider the theology, the philosophy question of how human interact with the world all involve brain interactions. And more recently to 19th century, there's a discipline called phrenology. This in Chinese means 颅相学, which means the scientists at that time, okay, about 150 years ago, they judge how smart this person is by judging the skull shape of this person, okay? So even though this is, seems to be pseudoscience nowadays, this phrenology impact neuroscience on the compartment of brain. So now we know that the brain, inside the brain, we have vision area, we have olfactory area, and we have central processing unit like this. This is actually inherited from this phrenology. And more recently, psychology actually became independent in late 18th century. Okay, so I think this is all relevant to brain science. However, if we really talk about modern neuroscience or modern brain science, I think most of the biologists will agree that this really starts from the era of Golgi and Kaha. Sorry about this phone call. This is one difficulty of online meeting when I speak in my office. Sorry, so I'm talking about that the real starting point of modern neuroscience really starts from these two gentlemen, Golgi and Kaha, who developed a new technology that enable human being to visualize individual neuron inside a brain. It's a dead brain, not in vivo, but they are able to reveal that the brain is composed of many, many neurons. Okay, so this is called inauguration of brain science. And these are the figures that Kaha drew. He was a very talented painter that he drew individual neuron inside the brain by labeling. The labeling is called Golgi label. So Golgi staining. This is a technique that some of the brain research lab still uses nowadays. Okay, so this was developed more than 100 years ago. So these are beautiful, beautiful neuron images inside an intact, inside a human brain. So after 100 years of Kaha and Golgi's discovery of human brain is made of a lot of neurons, let's ask ourselves, do we really understand our brain now? And so as I mentioned earlier, I think the answer to most of us is probably no, not really. The understanding of our brain is probably similar to what we know to the universe, or say similar to what we know to the bottom of the ocean. So I would like to share with you, in my viewpoint, what are the bottlenecks to prohibit us from understanding our brain. So one of the bottlenecks is that, as you see from Golgi and Kaha's work, they are able to draw individual neuron, including dendrite, axon, these fibers very beautifully. So these are called hardware connection of the brain. Okay. But if you think about to understand a computer, let's analog brain to a computer. If we want to understand a computer, it is far from enough to decompose a computer, get every IC out, and try to understand the circuit inside. Even if we understand all the hardware circuit inside a computer, do we understand how computer function? I guess the answer is again no. And there's a clear reason that for a computer to properly function, it requires accompanying software. So I guess this is similar to our brain. So let me define software, let me borrow the term software from computer science to brain science. So if we define software as programs and operating information used by a brain, then it is clear that currently we don't have any tool, we don't have any technology that allow us to see software inside a brain. To be able to pick into software, one possibility is to perform functional imaging in vivo. Okay, so this does not directly tell us what is a software, but it might tell us what is the consequence of a software functioning, and we might be able to back engineer to figure out what is the functioning software inside. Okay, so functional imaging in vivo is probably the first step to try to unravel the software inside the brain. And to perform functional imaging in vivo, it really requires contrast for information. And for brain information transduction, it uses voltage or ionic, like calcium, like potassium, ions inside the neurons. So first of all, we need to have very good contrast for imaging, for information imaging. And the second requirement is that because we need to see functional dynamics in vivo, so the imaging speed must be high enough to capture this voltage dynamics. The time span of voltage signal inside a brain is typically around millisecond, and the time span for ionic, for calcium, is typically on the order of 100 milliseconds. Okay, so if we want to understand the brain dynamics, functional dynamics, we need to have imaging speed as high as at least 100 milliseconds, or preferentially down to millisecond scale. On the other hand, it is also very, very important to understand the brain not only fast enough, but also with the large volume together. Why? Because for the consciousness, or say our memory, our feeling, they are all not come from a single neuron in the brain. They are based on the interconnection of many, many neurons inside the brain, and this is called emergent property. Okay, so let me emphasize this part again. To understand how a brain function, we really need to map the dynamics of many, many neurons simultaneously inside intact living brain to get understanding of the emergent property among these neurons. And this is the movie I'm playing here is a recent electron microscopy data of an intact whole brain. And for physics students, make a guess, what kind of brain is this? What is the animal of this brain? Okay. The answer might be surprising to most of us. This is not human brain. This is not mouse brain. This is Drosophila. This is fruit fly. Fruit fly is the animal that if you put an orange on your desk and live there for a week, probably you are going to see a lot of fruit fly coming around this orange, right? So this is the brain of this tiny, tiny animal, Drosophila or fruit fly. The diameter of this brain is about 400 micrometer. And please note that the time it takes to get the whole brain image with electron microscopy with 8 nanometer spatial resolution in 3D, this image takes a whole month to finish. Okay, so you can imagine if we want to use the same technology for whole human brain, it will probably take, I think I calculated some time ago, 1 million years to finish the experiment. So this is really not accessible nowadays. Okay, so this Drosophila brain is the most complete map of an animal brain nowadays with highest resolution. Okay, but having said this, please note that this is taken in electron microscopy, so this Drosophila is dead. Okay, this is not in vivo imaging, so there's no opportunity to capture the functional dynamics inside. This image is really a hardware connection again. Okay, so after 100 years from Golgi and Kaha, now we can do much, much better. Of course, the resolution is few nanometer, and the size, the imaging processing is much, much better than Golgi and Kaha. But still, scientists are pushing toward hardware connection. Okay, so to push toward functional imaging or say software mapping, I would like to say the second bottleneck is really to map the whole emergent property together, which means we need whole brain cellular imaging, at least cellular. Okay, for cellular, I mean at least individual neuron. Okay, and please note that this is actually a hypothesis. The hypothesis is that the function of the function of brain comes from the interconnection of neuron. So neuron is the basis of information processing. Please note that this is a hypothesis. Some of the neuroscientists believe the information processing is on the level of synapse, which is 3 orders smaller than neuron. So there are much, much more synapses with diameter about 100 nanometer inside a brain. So if we want to see whole brain synapse imaging, that will be even more challenging. So currently, we put our aim at whole brain cellular imaging, whole brain individual neuron imaging. Okay, so to achieve this, we certainly need enough field of view to cover the whole brain, and in particular, the depth is typically the most challenging direction. And in addition to that, we need subcellular resolution. Okay, so combining all this together, we need good contrast, we need high-speed imaging, we need deep imaging, and we need high-resolution imaging. Each of it is not particularly difficult, but if we combine all 4 requirements together, honestly, there's no technology nowadays that can achieve this in a human brain. Okay, so this is why called the bottleneck of contemporary brain science, why it is really difficult to understand the brain, because there are so many technical difficulties. So let me list all the imaging technologies nowadays, and the horizontal axis is resolution, the vertical axis is imaging depth. On the upper right region, these are medical imaging modalities like PET, MRI, CT, or ultrasound. These are typically having very good penetration depths, so they can penetrate through the whole human body, right? That's why they are used as a medical imaging modalities. However, their resolution is typically on the order of millimeter. So the size of a neuron is about 10 micrometer to 100 micrometer, and the resolution of these imaging modalities are still not enough to capture individual neuron inside a living brain. So on the other extreme, where high resolution is required, electron microscopy certainly offers the best resolution over the imaging technologies. However, the penetration depth of electron microscopy is really limited, not to mention that the sample has to be placed in vacuum for electron microscopy. So this is also not good for in vivo brain research. Okay, so if we look at all the existing technologies, the real bottleneck of brain research, as I briefly mentioned in last page, there's no existing technique that provide study of living whole brain function on a single neuron basis. Okay, so this is my viewpoint of why we are still not able to understand our own brain because of a technical challenge. And there are some development recently, for example, on X-ray imaging. The wavelength of X-ray is really short, so it might be able to provide a high resolution image because of the diffraction limit. But the penetration, but the issue of X-ray is really the dose issue. If we want to use X-ray to penetrate through the whole animal, typically we need to give enough dose to the animal, and partial part of the X-ray is absorbed during transmission of the animal. So this creates a damage possibility of the animal. And currently, Dr. Hu Yuguang in synchrotron is able to use X-ray to provide the image of a whole Drosophila brain with about 100 nanometer spatial resolution and 1 millimeter penetration depth, right? It should be straightforward for X-ray. But currently, this is limited, again, to in vitro brain, which means the brain is dead, which means the brain is fixed. It is still not possible to use synchrotron X-ray to do in vivo imaging for a brain, as I know now. Okay, but I'm really looking forward to possibility in this direction. Probably we are able to overcome the damage issue and use X-ray to perform very high resolution whole brain function imaging. So having discussed these technologies, I would like to share with you my viewpoint is that probably the best option for research nowadays is to use optical microscopy combined with a small animal model to be able to achieve whole brain function on a single neuron basis. So today I would like to introduce a technique called multiphoton microscopy and tell you why this enables us to improve the penetration depth and enable us to observe whole brain function at least on a very small animal. Okay, so this is the first part, why studying the brain. So now I enter the second part is how. So as the title presented, the method that we are going to use to study brain is to use light microscopy. And the reason is because light microscopy gives us the resolution of at least individual neuron or say subcellular resolution with adequate penetration depth into a small animal brain. Okay, that's the main motivation of using light microscopy to study brain function. So I guess every one of you have used optical microscopy, right? Think about your elementary school or middle school. I guess you must have used light microscope in those years. So very often the microscope you used was called bright field microscopy that is able to see a slice of sample, right? If you remember that in the old days in your school, you always use optical microscope to observe very thin samples, okay? One question I always like to ask students when I give a talk like this, have you ever placed your finger down to the microscope? Do you know what you can see? Okay, it's once again challenging for me to see your face. Well, that's the limitation of online format, but please make a guess. If you put your finger onto a microscope, onto an optical microscope, what do you see? Okay, and I guess probably some of you did this before. I also did this before when I was young, and the result is I don't see anything, right? But why not? So as a physicist, we always want to ask why. So I would like to invite you to consider two things. One is if we think about microscope, there are some important factors to consider. And let me say contrast, speed, depth, and resolution, which I already mentioned in the first part of the talk. And if I ask you to choose one, which one is most important? So this is a place that I would like to ask Junhui's favor. So please write down your choice, 1, 2, 3, 4, in the chat session, so that we are able to see quickly what did you choose. Okay, so there are 4 factors of optical microscopy: contrast, speed, depth, and the resolution. If you choose only one, which one do you think to be the most important factor? I would try to jump out.
Yeah, I see most people choose one. There's only one choosing 3, 2 choosing 2. All the others are choosing 1.
Ah, great. Many of you choosing 1. That's very good.
Yeah, 95% chose 1.
Thank you so much. Yeah, I hope this works well, even for online format. But anyway, wow, but that's really impressive because very often when I give the talk for typical audience, most of the person when thinking about microscopy, they choose resolution. Okay, so I think this is a very good starting point. Most of you choose contrast, which I cannot agree with you more. That I work in this field for about 20 years, and if you ask me to choose one, I certainly choose contrast. And the reason is really straightforward. No matter how fast is your imaging, how deep is your imaging, if there's no contrast, you see nothing, right? So the first important factor to consider is really contrast. And I'm very happy that we agree with each other on this part. And okay, so when I talk about optical microscopy, I mentioned that the best strategy is to use optical microscopy with small animal. And the model animal we choose is not human, it's not a mouse. It's the same as the electron microscopy data that was published 4 years ago. We use flybrain. The reason we use flybrain is because I think this reductionism is familiar with physics. That means to understand the nature of complex things, one of the best strategy is to reduce them to the interaction of their parts. In physics, we deal with this very often, right? We reduce a real planet movement down to a single point. Okay, so this reductionism is typical in physics and in biology. They also follow this reductionism. So to study brain, we start from this very small flybrain, and to supplement this idea, I also quote Nobel laureate Eric Kandel, who said that to bring the power of modern biology to study how the brain works, we need to study not the most complex, but the simplest. Okay, and I would like to emphasize that there's one very important thing preserved all the way from fly brain to human brains, that the signal transduction mechanism are basically the same. When I say signal transduction mechanism, I mean the ions that are involved in neuron signal transmission, the action potential shape, the action potential propagation. These are very, very similar across these different model animals. Okay, so certainly human brains are a goal, but before we are able to really probe the whole human brain, which is composed of 10 to 11 neurons, we start from a much simpler model animal. Even with this much smaller model animal, it composed of 10 to the 5 neurons, still a lot. And the signal, by understanding the signal transduction inside this 10 to the 5 neurons, we build up the functional connectome in this tiny animal, and we hope the mechanism can teach us how mouse brain and how human brain works. Okay, and so this is why we choose Drosophila as our model animal, and the main reason certainly small enough to be able to map whole brain functional connectome. But another very important advantage that I want to emphasize is that in Taiwan, we have largest neuron database among all model animals. And this was established by Professor Zhang Anshi in National Tsinghua University. So one of his major contribution to the brain science field is that he established the database of individual neuron inside a Drosophila brain, and established so-called standard brain model of Drosophila. This is very different from standard brain model of human being, because for standard brain model of human being, it is composed of different regions. Okay, but in this flight circuit database, the standard Drosophila brain is composed of individual neurons. Okay, so as I mentioned, inside a complete Drosophila brain, there is about 10,000 neurons. Sorry, 100,000 neurons, 10 to the 5, right? 100,000 neurons. And in this database, the latest released version contains 30,000 neurons. And there are still other 60,000 that are not yet published. So you can imagine that in a Drosophila brain, more than half of the individual brain has been mapped and constructed into a database, into a standard database. So this is a very, very important database if we want to understand the connection, the function of this tiny animal. Okay, so this is what I mentioned earlier about the strengths, the particular strengths of Taiwan in neuroscience is that we have this largest neuron database of Drosophila, and this is very important indicator of functional connection. I mean, when we perform, I'm the one who developed optical microscopy. When we perform optical microscopy to study functional connection, we refer to this structured database and make a guess which neuron connect to which neuron. This is the indicator of the functional connection. Okay, so this is really important of the world. And by the way, this is the only database that is able to cover more than half number of neurons. Okay, no such a database for mouse, no such a database for zebrafish, certainly no such a database for human being. Okay, so this is the only database that cover half, more than half of the neuron number in an animal. So that's why we do have a particular strengths around the world. Okay, so having talked about the animal selection, now let me share with you the challenges of using optical microscopy to observe tissues like Drosophila, like a living Drosophila brain. Okay, so as I mentioned earlier, if you put your finger on the white light microscope, what happened? Then typically you don't see anything. And I would like to ask you why. Why we cannot see blood vessels, why we cannot see muscle fibers inside our fingers if we put our fingers onto the conventional white light microscope. Okay, so the first option is because fingers too thick, so light cannot penetrate. And the second one is because white light is incoherent, so there's no clear images. And the third one is because optical scattering our tissue is very strong, light cannot focus properly inside our fingers. And the fourth one is because optical signals are generated everywhere in the finger, so the image is blurred. So once again, I need you to make a choice in the chat. And Junhui, please help me to take a quick look. Maybe I should also take a quick look. So now the question is why we are not able to see tissue structures inside our finger. Okay, the same question applies to brain. When we want to look at the brain, we want to look at whole brain, so we need to look into the brain.
You can see most people choose 4, and some chose, a few choose 1.
I remember I
3,
gave a...
Yeah, most people chose 4 at the moment, but second most is the number 1.
That's great. That's great. I remember now. I gave a seminar talk last semester, so this audience are the same. I guess that's why they know the answer, but that's very good. So you remember this. That's very, very good. I appreciate that. So maybe I can skip some of the technical details since most of the students know this. But anyway, I do appreciate many of the students understand this and or say you remember this. The real issue that it is very difficult to observe tissue is because for conventional light microscope, optical signals are generated everywhere. Why? And let me use a figure to tell me this. So if we think about fluorescence as an example, so imagine this is a sample, a tissue that is filled of fluorescent molecules, and there's a blue laser illuminated from the top. So you can see a focus cone here. Okay, this is objective, so there's a focus cone. So because this is a single photon absorption, like where I draw here, single photon absorption means the dye inside the tissue absorb 1 photon, emit 1 fluorescent photon. This is single photon interaction. So because this is single photon interaction, we can imagine that throughout the focus, everywhere inside the focus, fluorescence is generated, right? So if we look from the bottom or from the top of the objective, we do see green fluorescence, but the fluorescence emitted at a different layer are accumulated together onto our eyes. So it is not possible for us to distinguish which signal comes from which layer. So we see blurred image, and this is a muscle tissue image. And this is same for brain imaging. Okay, so if you put a brain, an intact real brain, under a microscope, you also see this blurred image. And the reason this image is blurred is because there are so many images that are generated simultaneously, and all these images are overlapped together. So when you see too many things together, effectively you see nothing. Okay, that's the problem of conventional microscopy. And I think 70 years ago, there has been an idea to overcome this limitation called confocal microscopy. And the essence is really to put a pinhole in a detection path. So this is a sample, there's a focus inside, and the pinhole is located at the conjugate plane of this focus. So only the emission from the focus goes very well through the pinhole. For the emission in front of the focus, it was blocked by the pinhole. For the emission behind the focus, it was also blocked by the pinhole. And this provide a property called optical section, and this allows us to see very clear optical sectioned image inside a tissue. So once again, this is a muscle tissue before confocal and after confocal. And the point I want to emphasize is that this means enhancement of actual contrast. Very often people misunderstood confocal as improving resolution. Actually not. It really improves actual contrast. Okay, so I hope this, you also keep this in your mind, that contrast is the most important factor, and to be able to see components inside a tissue, contrast is the most important factor. But certainly it is easy to imagine, it is easy to understand that the confocal microscope setup wastes a lot of photons because the emission is everywhere, but we only collect focus. Okay, so there are a lot of emitted photons wasted. So to improve the efficiency of this confocal imaging while maintaining the optical section, about 30 years ago, there was a technique called two-photon fluorescence microscope was invented, and this was published in Science. This paper has been cited over 10,000 times. Okay, this is really a cornerstone paper in the field of biomedical imaging. So the idea of multi-photon imaging is that there is a transition called two-photon absorption, which means the molecule inside this fluorescent medium absorb two photons simultaneously and then relax to emit one fluorescent photon. Because of this two-photon absorption, the incident beam and the emitted photon has a quadratic power dependency. Due to this quadratic nonlinear power dependency, only the focus emit fluorescence. Okay, so automatically, sorry, this dot is very small. You have to look into the figure to see this dot. I enlarged it here. So you can see that only the focal point emit fluorescence, nothing above, nothing below, only the focal point. Okay, so this gives us a very nice intrinsic optical sectioning capability, and this gives a very good tissue imaging. In addition to that, because the excitation wavelengths now become long wavelengths, okay, because of two-photon absorption, the photon energy of individual incident photon become lower, so the wavelengths become longer. The long wavelength excitation gives an additional advantage that penetration depth can be improved, okay, due to this near-infrared excitation wavelengths. So multi-photon imaging really gives us an ideal property of mapping individual layer by layer of a living Drosophila brain. So on the right-hand side, this is two-photon imaging. You can see we can visualize tissue layers one layer by one layer. And the left-hand side is wide field imaging. As we expected, everything is blurred, mixed together due to this wide field excitation nature. So this is the method we choose. So I remind you again, this important factor to consider. For now, I share with you, we use two-photon fluorescence to provide actual contrast, but because two-photon fluorescence only exists in one point, okay? So to see a whole brain, we really need laser scan, and this is a major limitation for the imaging speed. In terms of, and in addition, the achievable imaging depth is limited by wavelength choice and by attenuation in the brain, and the resolution is still limited by diffraction, okay? So in the following, I'm going to introduce what we did to push the limit of optical microscope to these limitations. And I would like to quote Sidney Brenner, who said that the progress of science depends on new techniques, new discoveries, and new ideas, probably in that order. So most of our research in my lab in brain science is to developing new techniques to push the limit of optical microscopy. So as I mentioned in the last page, the major limitation is in speed, in depth, and in resolution. So we developed several different technologies to address these challenges. One is speed. We develop a volumetric 2-photon microscopy. To improve depth, we use 3-photon fluorescence, and to get a better resolution, we use localization microscopy to break diffraction limit. Due to time limit, today I only introduce our main result on volumetric 2-photon microscopy because this really echo the topic of understanding the dynamic, the function inside the brain. Okay, so now I go to the third part of the golden circle what. So the result I'm going to show you is how is the functional connectome of individual neuron inside the brain. And the principle of this high-speed imaging is based on an interesting lens called a TAG lens, tunable acoustic gradient index lens. So let me start from gradient index lens. Conventional lens rely on optical path difference, rely on shape difference, but the index inside is the same, okay? But we all know that the focusing is due to optical path difference. So certainly we can create a lens with a fixed L, but changing refractive index inside. This is called gradient index lens. With this kind of lens, even the shape is rectangle light focus after this gradient index lens. And have the idea of green lens. Now let's think about generating an acoustic standing wave in a material. So when we have an acoustic standing wave, the density of the material changes periodically inside the material, right? And due to this density variation, refractive index also changes periodically in the material. So that means when we create an acoustic standing wave inside a material, we effectively turns this standing wave into a fast switching, focusing, and diverging lens, okay? And this periodic modulation can be as high as 1 megahertz. Okay, so at one moment it is convex lens, at the next moment it is concave lens. So when we combine this TAG lens together with objective, initially when the TAG lens is off, let's say the original focus of this objective is here, at one moment it becomes convex lens, so the focus move a little bit forward, and at the next moment it becomes concave lens, so the focus move a little bit backward. That means the focus move very fast on the order of 1 megahertz in space along Z direction. Okay, this allows us to do very fast scanning in Z direction so that we are able to see everything along Z direction almost simultaneously. Okay, this is the idea of combining this so-called TAG lens plus conventional laser scanning. So conventional laser scanning composed of point scan, arbitrary line scan, and a 2D frame scan. Now, when we add this TAG lens, a single point scan gives us an actual line, okay? And this takes about 1 to 10 microseconds. And when we have an arbitrary line scan in a conventional laser scanning microscope, by adding this TAG lens, this gives us a ribbon scan with arbitrary ribbon shape, and this time scale is about millisecond. And if we want to see the whole volume, so we combine this 2D frame together with this TAG lens, then we see the whole volume with a time scale of about 10 hertz. Okay, so this is a way we are able to visualize the function inside a whole brain, inside a volume of the brain. Okay, so to demonstrate this, the left-hand side is a conventional 2D laser scanning 2-photon microscope, and the right-hand side is a result of adding this TAG lens. So you can immediately see that with the same amount of scanning time, in this case is about 1 second, with same amount of laser scanning time, we see a lot of more details at a different depths. The color here means different depths. Okay, so we see all the details. This is olfactory circuit inside Drosophila brain, and we can monitor the whole circuit simultaneously under laser scanning. In addition to that, if we zoom in, we see that the left-hand side is conventional two-photon scanning by combining about 100 layers together. We form a Z-stack and we see individual neurons in 3D. However, on the right-hand side, this is the tag lens imaging. We also see individual neurons, and a very important difference is that the left-hand side image takes about 100 seconds, and the right-hand side image takes in less than 1 second. Okay, so there's a huge time difference to get the whole image. And the point here is that we didn't miss the information of individual cell, so we still are able to see individual neuron here. Okay, so we are able to monitor the whole brain of Drosophila. So on the top is the color map of depth. On the bottom left is the fluorescence imaging of a living Drosophila brain while we provide olfactory stimulus. And on the right-hand side, this is a delta F over F showing the functional unit inside the whole living Drosophila brain in response to olfactory stimulus. Okay, 嗅觉的刺激 olfactory. So this is the idea of we are able to mapping every spike from every single neuron inside a living Drosophila brain. Okay, so this is what we are able to do now. And as I mentioned earlier, we are able to perform this ribbon scan, and this ribbon scan really enables to achieve millisecond temporal resolution. So I'm going to skip a detail here because of a time limitation. But the essence is that because the speed now is about millisecond scale, so we are able to unravel unconventional signal transduction sequence in this mushroom body, which is a central processing unit of Drosophila brain. Okay, so sorry that I'm going to skip the detail, but I would like to emphasize that this highlight is that we are able to achieve 3D imaging within about 100 milliseconds and ribbon imaging within 1 millisecond. Okay, so that's why speed is really important, because this allows us to see the functional response of an intact brain. So one application example is the anterior, is a visual pathway. Let's say the vision system in the Drosophila has a strange phenomena that the eye of Drosophila is composed of about 800 small eyes called ommatidia, 复眼. Okay, ommatidia Drosophila has about 800 ommatidia, and this ommatidia signal comes into a region called AOTU, and this AOTU is composed of about 12 small compartments, and then diverge into a region called BU. This BU has about 80 microspheres. Each microsphere is about 2 micrometers in diameter. So no one knows why the visual signal transduction goes like this, converging into 12 subsection and then diverge into 80 microspheres. Okay, this is a nature design, but no one knows why. We also don't know why, but at least we have a tool to unravel the coding of this upstream AOTU and the downstream BU neurons. Okay, so the experiment we perform is called volumetric or optical physiology, which means that we are able to excite precisely one of the subregion in the upstream neuron and observe the whole volume imaging of BU. This BU is the microspheres is distributed around 40 micrometer depths. Okay, so we still need this volumetric imaging to capture all the dynamics. Okay, so this is a result. We are able to capture this 80 microspheres simultaneously and observe the response after precise stimulation. And this is called all optical physiology. Okay, so by stimulating region 1, region 2, region 3, we observe different microspheres have responses, and then we can plot the connection, functional connections of the BU versus the excitation, the stimulation of AOTU region. Okay, so this is called functional connectome in vivo. So with this technology, now we are able to establish 3D neuron functional connectome in vivo in arbitrary area inside a Drosophila brain. Okay, so this is what we can do now. So this is about technical progress. And more recently, because every time I give this talk, people ask why only work on Drosophila? Can we use this in mouse? So recently worked together with Professor Chen Shiguo in NTU in biological department to extend this volumetric imaging into mouse. And because the mouse brain is much, much larger than Drosophila, so certainly we are not able to cover the whole mouse brain at one time. Instead, we use endoscope. So we use a method called green. We use another green lens, which looks like a thin rod, and put this into a mouse brain and combine our volumetric imaging together with this green lens. So this enables us to observe the bottom of the brain called SCN region. This bottom of brain region controls circadian rhythm, 生理时钟, which means you every day wake up and sleep in a regular time due to this SCN region. And conventionally, the study of this neuron of this brain region is very, very challenging because this is located at the bottom of brain. So by working together with Shiguo, now we are able to observe whole SCN together with individual neuron response inside. Okay, so this is more recent progress. In addition to that, we also work together with an Italian group to combine adaptive optics together with microscope. And the reason I want to mention this is because adaptive optics might be familiar with our astronomy colleagues. As adaptive optics is commonly used in astronomy microscope to compensate atmosphere turbulence, right? And similarly in microscope setup, because we are looking into tissues, brain tissues. So the aberration caused by brain tissues is also severe, and adaptive optics help us to compensate the aberrations caused by these brain tissues. And we work with an Italian group, they innovatively develop plug and play adaptive optics, so which is conveniently combined with a microscope objective, and we can perform imaging directly with the volumetric imaging take lens. So the result is just published with the undergraduate student, Zhan Ling, and he did very well to demonstrate contrast enhancement with this plug and play adaptive optics. So another ongoing project is that now we are able to achieve even higher volumetric speed by combining multiple ribbon scanning together. So this is carried out by two of my master students. So in essence, we split one laser beam into 32 beams and using a Galvo mirror to scan across one direction, and the tag lens give another fast scan in Z direction. So equivalently, it forms a volumetric imaging with this 32 ribbon scanning, and the speed can be as high as 500 volume per second. So we can use that to probe high-speed flow cytometry with individual microspheres visible, as well as very, very high-speed functional imaging inside a living brain. Okay, so I'm going to skip the details. One very interesting recent progress is that Jensen developed a machine learning code that help us to convert the high-speed, very noisy image into the ground truth image, similar to the ground truth image. So we are highly looking forward to the new development in this direction by combining high-speed microscopy and machine learning. So in summary, I would like to echo again why we would like to study brain, because this is really fundamental in physics, and this is really challenging, okay? But the other side of challenging also means there are many, many exciting opportunities. So I would like to invite and encourage students as well as our faculty members to join these challenging and exciting research opportunities. And the method we choose is optical microscopy because it provides penetration depths enough to a small model animal like Drosophila, while having subcellular resolution that enable us to map functional response of individual neuron inside a living brain. So we are now pushing the limit in terms of speed, depth, and the resolution. And today I share with you volumetric two-photon microscopy, and they are also deep breakthroughs in depths, also in resolution that I have no time to show, but you are welcome to find me and give more discussions. So regarding the what, the ultimate goal of this optical microscopy imaging is really to map the whole brain functional connectome that will enable us to answer the question how a brain functions, okay, even in this very small, tiny animal. And then we probably will be able to transfer the knowledge into the function of mouse brain and the human brain, okay? So I would like to also advertise a little bit to the Brain Research Center in National Tsinghua University, because originally, I mean, when Junhui invited me, I actually contact Professor Jiang Anshi in NTHU, because I think he is the perfect candidate to deliver his talk, but unfortunately he cannot make it. So I would like to mention that in Brain Research Center of NTHU, well, he always want to say Brain Research Center is not only in NTHU, it is really Brain Research Center of Taiwan, okay? So they are developing several innovative technologies. As I mentioned earlier, new technologies really drive new ideas and new discoveries in brain science. So in addition to functional imaging I just mentioned, they are developing whole brain super resolution imaging, and they are developing synchrotron X-ray microscopy for the whole Drosophila brain high resolution mapping. And their database really provide a very good guidance for memory connectome inside Drosophila brain. Okay, so there are various new technology, new exciting technology development in Brain Research Center. So in the end, I would like to share a few open questions to stimulate interest in our students and faculties. One of the open questions, now we are able to see, as I mentioned, we are able to see functional response across the whole Drosophila brain. But how should we link this functional response to software? Okay, this part I haven't found a clear answer, and so that's why we are working with some expert on neural network simulation to trying to figure out brain software. And I recommend interested students to look into this paper in PLOS Computational Biology. It was two neuroscientists who are interested to know if they use modern tool in neuroscience, can they understand a microprocessor? Okay, so this is an interesting work, and you're welcome to take a look at this. But one of the questions now in brain science is that how to derive software. And another open question is how can we apply this technology to human brain? Okay, so one endeavor that Elon Musk is working on is really to use so-called brain chip to put in human brain. So here is a very short video showing the monkey experiment of this brain chip. They are able to put chip inside a monkey brain, okay? And this chip have about 4,000 electrodes, and it records the electrical response inside this mouse brain while the mouse is playing game, okay? So when they record and analyze the response inside the brain, eventually they feedback to the control, and you can see that the monkey can now play games without using hand. The monkey is controlling the game by his brain directly. Okay, so this is really something amazing to perform. But even with this brain chip, they only use 4,000 electrodes, while in a human brain, as I mentioned, there are 10 to the 11 neurons. So is there any possibility that we are able to map the whole brain? Is still an open question. Okay, one of the last open question I would like to raise is a bold one. As I mentioned, in the 125th anniversary issue of Science, the #1 question is, what is the universe made of? And I think now we know that there are majority of the universe, the majority of universe is composed of dark matter and dark energy, which we really don't know. And the second one is, what is consciousness? What if they are relevant? Okay, so I made a quick map linking consciousness in the dark matter and the dark energy. Why? Because if we think of consciousness as kind of software, this software is composed of information. And it is well known that information possesses entropy. And recently, there was emergent theory saying that entropy is able to derive gravity, as well as entropy is able to derive Coulomb force. Okay, so this provides attractive force, this provides repulsive force. This might link to the dark matter and the dark energy. Okay, so I have no certainty on this part. This is a bold guess, but somehow I feel the information entropy might lie in the center of linking consciousness to our universe. So in the end, I would like to acknowledge the collaborators in this brain technology project. So currently I have a brain project in MOST. So there are many collaborators in terms of technology development, in terms of signal analysis, and in biological development. And we also work extensively with a lot of international collaboration. So I would like to appreciate these collaborators. And most important of all, I would like to thank my students who always work very hard to deliver nice images in the lab. And I'm the one who represented them and give a presentation here. So thank you very much. And you're welcome to explore exciting neuroscience together.
Okay, thank you. What a great talk. Now is the Q&A session. So for those who are in the meeting room, you can actually raise your hand and then speak up. And for those who are actually in the webinar room, although you cannot talk, but you can type, I will try to read out your question that you type in the webinar room. So now is the time for questions. Anyone who has a question can speak up. Remember to unmute yourself. Now everyone is muted. Yeah.
Maybe let me try to see who is asking. I still prefer to see face to face.
Any questions? Oh, there is one question from Chen Qunwei. He said human brains are much bigger than the fly brains. Can they still be studied using optical methods? I think this was actually Johannes' work, right?
Yeah. This is a very good question. I should say all the endeavors in Drosophila brain imaging, Drosophila brain study, finally should lead to our understanding of human brain. Okay, I think this is the ultimate goal. Everyone working in brain science understands this. However, the technology barrier, as I mentioned in the talk, really prohibits us to study whole human brain simultaneously now. Okay, it is possible, it is indeed possible to study a brain region with optical microscopy now, but for human brain, there's another ethical issue. But if we skip the ethical issue, even if we are able to map a functional connectome with optical microscopy, now in human brain, the only accessible region is probably about 1 millimeter cubic inside. So this is still very limited to allow us to understand the whole brain, but it is good enough to allow us to understand individual brain region. For example, amygdala. Amygdala is 杏仁核, for example. Yeah. So this kind of technology still provide potential to better understand and better study human brain. This is my viewpoint, and we are still waiting. We are still looking forward to emerging new technologies that will probably enable us to visualize the whole human brain with, say, micrometer spatial resolution. Yeah, if you are able to do this, you certainly win Nobel Prize. Definitely. So I should share this information to students, right? If you're able to develop a technique that allows us to see the whole human brain with micrometer, no need to be super resolution, just 1 micrometer, whole human brain. This will certainly be Nobel Prize winning technique.
Okay, more questions? Anyone who want to speak up? If you want to speak up, remember to unmute yourself. Yeah, the button is it's a button left corner. You have to click the button to unmute yourself. If there's any technical issue that you couldn't speak up, you can always type, in the dialog box. Oh, very quiet here.
That's the difficulty for online.
Oh, there's someone who raised a hand. Yeah, you can just speak up. You are only one who raised a hand, so just speak up. Yeah.
By consciousness, did you mean that the universe think stuff itself, or mean the organisms think and the universe functions?
Do you want to ask the definition of consciousness, or you want to ask the relationship of consciousness and universe? Which one you're asking now?
Yeah, I mean, what is the consciousness you mean in the very last slide?
Okay. Good. The consciousness I refer to is the consciousness in our brain. So, well, let's say in this way, the definition of consciousness is actually also in controversy, but let me use a common definition of consciousness is you exist. How do you know you exist? This is called consciousness. You have an awareness of you exist, and you have an awareness that there are things surrounding you and they interact with you. This is called consciousness. Okay, so when I refer to consciousness, I refer to this kind of general definition of consciousness, and this is the issue that haven't been really understood in science. What is the consciousness? What is the origin of this consciousness? As I mentioned, as I cite the science spatial issue, this is one of the top questions that scientists is eager to figure out. But even after 15 years, the spatial issue was in 2005, right? So yeah, I should say already after 15 years, I think we still don't have a consensus on what is the origin of consciousness. There's no general consensus yet. So that's why this is an area worthy to devote your life to, because no one knows the answer. So probably you're the one who figure out how it works.
I see.
Hopefully this answers your question.
Yes, yes. So basically, I was wondering what is that consciousness mean? So we need to know that first before we can have a relation between consciousness and the universe.
Certainly.
So I ask you.
Certainly. So as I placed in this last page, this is a very bold link between consciousness, because there is a famous theory called information consciousness theory. So you can check this online, and the basic idea of that theory is consciousness is composed of information. Okay, so and my idea about software. So consciousness is like a software. This is my idea. But no matter software or information, well, software is also composed of information, right? So what I try to link is a very strange, very bold link to link information with consciousness, and the link information with entropy. This part is okay. Information with entropy is certainly okay. But then in entropy to the universe. So this is a very big link, and I am not sure, well, I'm certainly not sure this is correct, but based on existing literature, each arrow in that model exists. Each link in that model exists. Okay, so that's why I think probably this is the overall picture we may work on in the future.
So do you think we have already reached the limit with the current methodology using this kind of optical imaging in
No,
terms of no, we haven't. So you think technology-wise, there's still a lot of room for improvement?
yeah, there are new technologies coming out every year. Yeah, you know, there was a new journal, not really new now, but about 10 years ago, when there are a lot of new journal coming out in the series of Nature journal, there's also a journal specifically devoted to nature to method development called Nature Method. There are a lot of new optical imaging technologies coming out every year.
With a higher resolution, better contrast, that kind of improvement.
Exactly.
Yeah.
My feeling is that within 10 years, we will be able to map a whole mouse brain with individual cell resolution. We are not really far from there. Now the best technology gives us about 4 millimeter penetration with cellular resolution, and the whole mouse brain is about 6 to 7 millimeter. So we are not really far from there.
Okay.
But whole human brain is another story. Okay.
So how many more students do you need in your lab?
That's a good question. How many more? I don't know a specific number, but well, we always hope to work with motivated students. Let me put it this way. It's really it really doesn't matter whether or not you have optics background or you have neuroscience background. That doesn't really matter. What we really is the motivation. If you feel this is really interesting, really challenging, you are welcome to talk with me and learn during your exploration.
I totally agree. I mean, this is better answer than counting the heads. Yeah.
Yes, of course.
The motive is more important. Yeah, I totally agree. Yeah. All right. So any more questions from the audience, either students or the faculty? Let me count to 5, 4, 3, 2, 1. Okay, so there's one. Okay,
there was a student asking question on the 讯息.
Oh, I see that. Yeah.
The message. Okay, he typed here. Hi, Professor. I would like to know if there is any successful implementation of reinforcement learning in the analysis of brain function or image quality enhancement.
Yes, yeah, that's a very good question. As many of you knows, deep learning is really a mainstream technology recently. So there have been a bundle of high-profile research papers on using deep learning and microscopic image contrast, resolution, as well as imaging depth enhancement. Okay, so you're welcome to find me, and I can give you a bundle of reference on this part. And if you're interested in working in this field, that's even better.
Okay, it's a good one. Any more questions? Right. Oops, someone is ringing.
That's my computer.
Oh, okay. Okay. All right. So if there's no more question here, then let's call it a day. So let's thank Professor Zhu again. Yeah, maybe you want to turn on your microphone to clap your hands.
Thank you so much.
Right. Thank you. Okay, see you next week. Next week, we are having a distinguished professor, Li Yuanpeng, talking about astrochemistry. Yeah, should be a very interesting topic as well. All right, so see you all next week. All right. 拜拜。
杨立忠跟姚玉琪没有到吗？
助教现在开始点名，所以请大家先不要离开。
杨立忠跟姚玉琪是没有到吗？没有看到的人，确定一下。就没有啦。那抽点，要抽点吗，老师？
你随意啊，你随便点几个，按照惯例。
哦，那
因为这
那
这边我大概随便抽几个在魏炳拿这边，你那边是讨论室，你可能看不到。对，那魏炳拿这边我随便问几个。
那萧守燕在吗？
在。
陈敬豪在吗？
在。
刘志轩在吗？这边应该是另一边讨论室的，没在。吴宗尧在吗？
在。有声音吗？
有。张佑明在吗？
肖志轩在我这边。肖志轩在。
张佑明在吗？
是聊天室
看到了。
打字说在。
梁如清。梁如清在吗？就这样吧。就这样。下课。
这边我点了一个，在。随便抽好了。
下周一样是线上吗？下周一样是线上吗？应该是吧。
下周极有可能是线上，要看学校政策有没有改变。学校政策如果没有改变的话，那么下周应该还是会持续线上。对，所以这一样是